I'm Charlotte Nelson. I'm from UCSF. I'm a graduate student in the bioinformatics program, and I'm in Sergio Baronzini and Atul Bude's lab. And today I'm going to be talking about big data and how we apply it to healthcare. So when it comes to big data, I feel like as a computer scientist, it's easy to just go out and start coding. And so it's really important for me, at least, to have some type of structure. And so I like to think about the DIKW pyramid. And so at the base of this pyramid is the data. So that is just, data by itself is just numbers, doesn't have much meaning to it by itself. But when we add context, we can then organize the data and it turns into information. Adding experience to the information turns it into knowledge. And then at the top of this pyramid, we have wisdom, in which we're at a state of awareness where we can actually combine our knowledge together to see a situation as a whole and make appropriate action. So why is this important? Well, we have a ton of data. And this data has mainly been collected in the last few years, and I think that that makes sense when you think about how data storage has evolved. And so the easier it is to store this data, the better we're able to collect it. So who has the biggest problem in big data? Astronomy, finance, e-commerce, or biomedicine? I'd like to argue that biomedicine is at least up there on the list. And the reason I believe that is because of the hierarchical organization of complex organisms. So if we just start at this bottom level of genomic information, we don't just have our own genome, but we have the genome of the bacteria and viruses living within us. This is transcribed into RNA and then translated into proteins. Proteins interact with each other and create complex networks and pathways. And then we have the behavior of the cell as a whole. An arbitrary amount of steps later, cells assembling into tissues, tissues into systems, and then systems into the organism. It's at that top level that we're observing the phenotypes like eye color, hair color, or even complex diseases. And so we're collecting data at each one of these different levels. And I think the crazy thing is that traditional research is just looking at a single one of these levels and trying to correlate it with a single phenotype above. And we do that without considering any of the relationships between these levels. And so thinking back to the DIKW pyramid, I think that it'd be better if we think about this all as a whole. So to try and approach that problem, in the Baronzini lab, Lily Wang started with ICT Net, and this is a powerful plugin for Cytoscape, right there. And basically, she took together a bunch of different databases with tissues, drugs, phenotypes, and genes, and combined them together for this resource. And this is just a very simple example of what you can do with ICT Net, but above we have genes, and below are diseases. The green circles are immune diseases, the red circles are neurological diseases. And so having this network, you can already start to see different patterns. And in this network, you can also see that similar diseases are clustering together. So now we're focusing in on the immune-related diseases. And then if we just look at some of the autoimmune diseases and reorganize the network, we'll find that the three most common autoimmune diseases of MS, RA, and type 1 diabetes up here actually share a lot of susceptibility genes. And so this is just an easy application using ICT Net. Moving forward, Daniel Himmelstein created HettyONet, and this is a much larger network. He brought together 19 publicly available high-throughput resources and combined them together in a heterogeneous network containing 22 types of nodes and 24 different types of edges. Now with this, the size and heterogeneous nature of this network, a lot of the traditional network analysis tools weren't able to be applied to it. And so he turned to social networks like Facebook and decided to use a method called degree-weighted edge count in order to try and predict new relationships between diseases and genes. And then because our lab focuses on multiple sclerosis, he looked at that. He was able to find 4 new novel genes associated to multiple sclerosis, and 3 of those were actually validated in GWAS studies. So the next question we had was, can we actually apply this to drug repurposing? So we deleted all the edges between the diseases and 1,500 small molecules that are FDA approved and tried to find the missing links. And it turns out that we were actually quite good at predicting known disease-modifying therapies. And even when we looked at clinicaltrials.gov, we still were able to prioritize these drug relationships. So since then, HettyONet has evolved into SPOKE, or Scalable Precision Medicine-Oriented Knowledge Engine. And SPOKE is now a multi-lab collaboration here at UCSF. It has since grown from just these nodes and edges, but we have pathways, molecular functions, biological process, cellular components, genes, proteins, compounds, pharmacological classes, side effects, diseases, symptoms, and anatomies. And I should mention that WikiPathways is part of our pathway node type. And it's really great now that we have this large team that we can actually keep SPOKE up to date. We update all the nodes and edges on a weekly basis, and it's just growing very fast. So what's next? Well, I mentioned I'm also in a Tool Buttes lab. So that plus the UCSF Precision Medicine Initiative, I thought that it was fitting that we would take all of the patients at UCSF and find a way to connect them to SPOKE. And so my idea behind this was that we could take the electronic health records, or EHRs, and take all the different codes and concepts, and then find those same codes and concepts in SPOKE. We could then move from those original concepts out to additional nodes in SPOKE, then producing this barcode for a given patient at a certain time point that really describes the patient in terms of the nodes in SPOKE. So what this looks like in practice is that before we can actually look at an individual patient, we have to learn what does a code or concept mean in SPOKE. So for that, we take, well, let's just say the diagnosis code for multiple sclerosis. So we take all of those patients that were diagnosed with MS, and we get all of their codes and propagate them through SPOKE, therefore learning what multiple sclerosis means to SPOKE. We then get the next thing, say it's drug X, we get all the patients that were prescribed drug X, do the same thing, and we just repeat this until we've seen every single code and concept in the EHRs. And then once we're done with that, we can actually combine this information together to understand the patient as a whole. And so we're left then with a vector for this patient, and the elements of the vector describe all of the nodes in SPOKE. So now we're really looking at the patient as a whole. Almost every level in that hierarchy I showed earlier are being represented in this vector, so we have a deeper understanding of this patient. Once we've done this for all the patients, in order to create a visual representation of them, we decided to organize the SPOKE nodes with a self-organizing map into a two-dimensional matrix. And that gives us a visually distinct representation for each patient. So at the end of this long process, we're left with over 300,000 patients with patient representation. Here we're looking at probably about 500 different patients at UCSF. And you can already see they each have a unique signature in SPOKE. So what can we do now? Well, now that this whole pipeline is set up, we can take anyone with a medical record anywhere. They don't have to go to UCSF, they just need to have a medical record. And so we take this group of PTSD patients, and there's about 4,000 of them, the records, and in seconds you can have these profiles. The cool thing is now we can start to ask questions about what these profiles actually mean. So this is the composite image for the PTSD patients, and we can ask what are the most similar diseases to PTSD. We see things like bipolar disorder, nicotine dependence, schizophrenia, alcohol dependence, panic disorder. And we can ask what is the least similar, and we see it's atopic dermatitis. And we can see that the PTSD signature shares a lot of hotspots with these diseases down here, but is almost the polar opposite of atopic dermatitis. And so even cooler, though, is that we can then ask what are these hotspots. And so these are the most, significant hotspots for PTSD. And we can actually pull out the spoke nodes that were responsible for that intensity. And if we zoom in, we see nodes such as depression, rage, migraines, suicidal thoughts in different areas of the brain. But we also see genes and pathways. And now we can go look into those genes and pathways and actually try to get a deeper understanding on what's happening with our patients. So that was just one application where we have a bunch more in the pipeline, but here are a few: disease pattern recognition, disease prediction, patient stratification, multiple sclerosis disease course, and drug signatures. So that's it. I'd like to thank both my labs, the Spoke team, UCSF Precision Medicine, and my funding.